Okay, so welcome to this research part of the talk. So basically, I gave a bit of intro to all the possible things, more or less, which we need to understand this part of it. So just to give you an essence, when we talk about systems biology, we were kind of, we know that we will try to somehow relate experimental data with and try to understand that using mathematical models. And often there will be, dilemma between different kinds of strategies. Say one way I can start with some experiments and then predict something, analyze it, and then see that whether how that in terms of some network topology, in terms of mathematical model makes sense, and then somehow reproduce those experimental data and try to predict something out of it, and then get ideas for new experiments. That is one way to do. The other way around is that what we have shown you earlier, that earlier people have proposed a mathematical model of certain phenomena, then they analyze that, they predict something, and later experiments came in and somehow showed that those mathematical model made sense and we can do something. And nowadays we normally go back and forth between these two models and experiment simultaneously to train the experiment or to train the model, vice versa. So there are three different strategies. Okay. Now, as I said, there are three different strategies. Is always a egg-chicken problem. That what to do? Should I do model first? Should I do experiment first? Or how I strategize? So in our lab, what we right now have started after 5, 6, 7 years, for the last 5 years maybe, that how we can utilize single cell level studies and understand cell cycle regulation by in terms of proposing different mathematical models, which is tuned to that kind of experimental studies. Okay. So to start with, I will try to give you something that relates with kind of a strategy one, where we had made a model beforehand, okay, and then somehow got some intuition, and from that intuition got some idea of experiments, and we try to validate our intuitions by experiments. So earlier I talked about that cell cycle regulation was important, and that G1 is, or this restriction point control is tightly regulated by this RBE2F control mechanism. However, in many cancerous cells, we see that RB either mutated or non-functional in many cases. Okay, so we ask the question that how restriction point gets controlled in different cancerous cells where RB is not that much functional or not working at all, and can we alter or control the restriction point? If there exists something like a restriction point, can we control that by doing experiments? That is one of the goals. The other one, where we begin experiments by doing some fancy experiments, got some intuition about how cells are growing under the dish, and have some interesting probe to actually calculate what is the length they are spending in the G1 phase and SG2M phase, and when they are transiting and when they are dividing. And when we calculate those, we see that there are heterogeneities in their cell division time, in their G1 time, in their SG2M time. And what are the origins of this heterogeneity? What is actually causing this heterogeneity? Both at the population level and also at the lineage level, those correlation things that I was talking about. And if we understand that, can we somehow control the heterogeneities in their cell cycle dynamics to get some fruitful output out of it? Okay. And finally, although I will not talk about that in greater details, what we have seen is that if we give them different probes, say for example, cells under different phases of their cell cycle, move or have a different growth pattern and different movement pattern, and that gets enhanced when we give certain other factors. Now that must have an influence in the case of wound healing and other things. So first, what we want to understand that how cell cycle and cell movements are kind of correlated there, and if we understand that, can we precisely control those to get some good output in terms of, say, wound healing or something like that. So those are the major 3 things, and there are other things also that we are working on. I'm not going into that, but I will try to at least I will try to explain these 2 and touch upon on the third one, hopefully. So let's come to the first problem, and I have already created the premise of the first problem, is that we have already seen that Yao et al. How beautifully they have performed their experiments to say that this RB2F control in and around the restriction point is being done by a switch-like mechanism. Okay, and you can see that if I go on changing the growth factor levels, I can see unimodal, little bit bimodal, then bimodal, then bimodal is going off, and then kind of an unimodal response. So it's basically E4 means it's a particular clone. So we have to, in this kind of studies, you have to make several clones and show that it is happening in different clones also. That's the reason. So this is fine. Now we are talking about where the RB regulation is not that much effective. Now if RB is not effective, what people have seen that there is a set of microRNAs. MicroRNAs is nothing but small nucleotide RNAs, about 22 to 24 base pairs. And earlier people used to think that they have nothing to do. But now people know that these microRNAs control the mRNAs of different important regulators, either by degrading them, by first making complex and then either degrading them or reducing their translational efficiency. Like E2F mRNA, it will bind and it will reduce the translational efficiency of E2F. On the contrary, E2F actually activates few of those microRNA 1792 cluster components, especially microRNA 20A and microRNA 17. And also we know that if I allow growth factor, MYC comes in and mutually activates the E2F transcription, and E2F can also activate its own transcription. And obviously sometimes RB is not completely mutated but partially there, or is not there at all. And the important part here is that if you give them growth factor, depending on the level of E2F, how much E2F has gone up, whether it has gone up here or here or here, depending on that, cells will show different outcome. If it has gone to a certain extent, it will cycle. If it has gone to a very high extent, it will opt for apoptosis. If it is intermediate zone, it will be going to the worst cancerous zone. If it is not at all active, it will be in the arrested state. So all possible features can happen depending on the E2F level. And that can be controlled by microRNA 1792. So this is what happens in absence of RB, or when RB is not that much influencing the RB2F control. Make sense? So if that was the case, that was not that much interesting. What was interesting is the following. What we observed in the literature is that if microRNA is an inhibitor of E2F, we expect that if the microRNA is overexpressed, cancer should not happen because E2F will not go up high. E2F will always be in the lower state, and E2F should be such that cells will not cycle. And if cell is not cycling, cancer should not happen. However, in many solid cancers, people found that in many I think I have done it opposite. So basically in many solid cancer, what has happened, that people have found that these are highly expressed and still cancer is happening. On the contrary, so just take it opposite, in the hematopoietic cancers, people have seen that these expression levels are low for these microRNAs, and the cancer was still happening, which makes sense. That microRNA 79 is not there, E2F will go up high and then cancer will happen. But it does not make sense in solid cancers or many other cancers where microRNA expression levels are high and still cancer is happening. So we made around 2018, we made a similar Tyson-like model with some phenomenological term of this network that I have shown here, and what we have shown is the following. So I have this overall dynamical regulation in terms of mass OD based mathematical model. I can do a steady state analysis where what I'm trying to follow is the total E2F expression level as a function of growth factor, And think about, I am doing it, so this KDMP is nothing but the how microRNA degrades, at what efficiency it degrades E2F. That is my only kinetic parameter. Right now, all other kinetic parameters are fixed, and this is also fixed at this value, a certain low value. And I get this kind of a wild type diagram. So the middle one is my wild type diagram. So it's a bimodality. So the model tells that it happens in a switch-like manner, still in absence of RB. Okay, makes sense. What happens if I downregulate or overexpress? If I downregulate, I see that the saddle node 1 moves towards the right side. That means the cells will transit towards cancerous state at a higher growth factor. So they become less cancerous. Right? If I need more growth factor, and I can still see that I am not going up to that level of E2F, which can cause cancer because it is even below the cell cycle level. So I will not be in the cancerous state, kind of. So if I move away, if the saddle node 1 moves away from the wild type scenario towards the right side, I will have a less cancerous situation. Okay. But if I overexpress the microRNA, I can see that saddle node moves towards the left. That means the cells will respond even at a lower growth factor and even go to a higher level of E2F. Make sense? Yeah, so basically if I think of, say this, if I think of this is a normal healthy cell, this is the level of E2F should be maintained for the wild type, is my cell cycle, normal cell cycle will be maintained. If the E2F level goes beyond that, I will be more cancerous, and if it goes too high, I will be apoptotic. If it is go below that, I will be not even cycling based on the E2F level. I am transiting all the time to the higher steady state from the low level with respect to growth factor. And if I transit at a lower growth factor, that means my propensity is too high to become cancerous and to go to a higher E2F level. Okay, makes sense? This up to this part. So now what we did, we kept all the parameter condition fixed. We just changed the efficiency level, that how microRNA degrades E2F. We assume that this is higher in case of the other cell type, maybe some another cell type, where I have my wild type scenario is something like this. But now you can see, if I overexpress, I go in an opposite direction. So earlier when I was overexpressing, I was going in this direction. My saddle node 1 is moving towards backwards in the left hand side. Now if I overexpress, my saddle node 1 going towards the right hand side, and it is basically annihilated. And if I downregulate, then I see the even greater cell cycle abilities, and you see the E2F levels are also going up higher at a lower growth factor when I'm downregulating now. So it works like kind of a magical thing, that only a single bit of that, only how the microRNA, how it is degrading E2F, just because of that alteration from cell type to cell type, cells can behave differently. That was the proposition from the model. Make sense? Some of them, some degradation parameter rates are from literature, other than we have curated. However, we are trying to recreate the phenomena. That was correct. That is true. That is true. The one way to do is like let's do some experiment and try to see that whether that makes sense or not at all. That's one way to go. Okay. So we thought that we will try to validate these two. So what essentially the model is trying to predict right now is that microRNA alters the E2F level and the growth factor threshold for the E2F activation that we have seen, the growth factor threshold, and by upregulating or downregulating microRNA, we can change the proliferation propensity. These are the two things that we right now can predict from the model right now. Let's see. Okay. So now we try to go in this, think about this cell line, for example, something like that. And we are trying to do a very similar kind of experiments. But now if you look at We got this reporter system from Yao et al. Like I have a E2F promoter and I have a D2GFP that we took it from Yao et al. They have given us. And what my student Vinodini made is like earlier, this E2F reporter cannot sense the intrinsic microRNA that is present within the cell because it only degrades at the rate at which the D2F-GFP degrades. What we did, we cut that degradation domain and we put the 3' UTR of the E2F such that now it can sense the internal microRNA that is present within the cell type. Make sense? Okay. So this reporter also we made, and obviously we made clones of this. And this is basically the microRNA 20A which sense and degrade. MicroRNA 20A is one of the microRNA 1792 cluster component which degrades or E2F. Okay. Yeah.
This is just reporting the promoter activity.
This is a promoter activity. Yeah, that,
Endogenous levels of
nothing. Nothing. Right now, nothing. That's a promoter activity. Yes. So what we see? What we see if I use this reporter in MCF7 cells. So right now, this reporter does not have that 3' UTR domain. Okay. And what we see here is that we start with low serum concentration and move ahead, and then we start seeing some bimodal scenario, but which are kind of tightly close to each other. Okay. Now, when we do a serum pulse experiment, just like Yao et al., and we come down to a lower level, we see that it starts like this, but the bimolarity more or less get extended till 0.1, like a very low level of growth factor even. So if I give them a burst of 20% and then remove the thing and keep them up to 24 hours, it looks like that. However, this does not sense the internal microRNA. So you have to think like this, that as if the cell line intrinsically does not have any microRNA, my E2F levels should be bimodal. Make sense? I do not have any microRNA in the cell, if that is the case right now. So basically it is not reporting the internal E2F activity because the internal E2F can be manipulated by the internal microRNA. And internal microRNA is present. But right now, this reporter cannot sense that. And that's why I'm seeing a bimodality right now. I don't know what will happen if I do the experiment with this reporter, the other reporter that I showed. At 20, okay, this basically I started with a very low serum concentration, right? So all the cells are synchronized in the early G1. So I kept them in a very low dose. So everything is synchronized. I have a single unimodal peak. Okay. Then I allow them to, for 24 hours, I give them these individual dosage, whatever dosage is, for 24 hours. However, I give them some hydroxyurea so that they cannot cycle. They will get stopped after transiting G1 to S. After that, they will stop. So that I pick them. And then obviously, right now I'm not showing you. We will also see the, what is the DNA content. And it also shows like bimodality. So you know that this is basically replicating the E2F dynamics and the DNA dynamics is kind of pretty similar. Something like that. Okay. Now, what happens when we do with another clone having this reporter where 3' UTR is there? When the 3' UTR is there, we see that this peak slowly shift from the left hand side, but without showing any bimodality right now. Okay? Same happens if I do a pulse down or pulse up, does not matter, it shifts. So essentially what it tells right now is that as if this diagram is kind of the scenario. So already my microRNA levels are such that my E2F level is kind of doing like this. And if I somehow downregulate my internal microRNA, I should be able to see something like this, because this is the evidence. This is the evidence that where my microRNA are there but cannot degrade my reporter. My reporter will be there, but if I now downregulate my microRNA, which cannot degrade this, I should be able to see this. Something like that. Make sense? I will slowly go back. Yes. Yes. So I don't know whether it has a high KDMP level. KDMP is a parameter, it's not a level. So what we know is that if in my MCF7 is well known, it is highly expressed, and if you downregulate, MCF7 does not get cancerous. That is known, that is experimentally proven, but on a different experiment. Okay. So what I am saying is that it will get more, if I downregulate, it will become much more cancerous. And that is known experimentally. However, whether it will go just linearly go this side or it will go a switch like this side, this model tells you that it will go like switch like. An experimental model also says that it will go switch like back. Okay. If I downregulate systematically, we will come to that. Now, just to make a sense, obviously MCF7 is not like RB null. There is RB. MCF7 RB is there. Now, what we try to see is that by doing a model where I have a tight high RB regulation and I have very placid RB regulation, RB is not tightly controlling E2F, I can get these two kind of a bifurcation diagram and the corresponding stochastic simulation. If I perform a stochastic simulation of this network where RB is there and microRNA is also there, what I see roughly, don't try to compare these to that, what you should compare is the following. If you see there is a high RB control, the peaks are quite far apart between the low and high. However, if there is a less RB control, you see the peak shift is kind of like this, like it shifts like from left to right in a continuous manner, but it shifts. So it tells you that it is a low RB control. However, it is microRNA is also controlling like that. Now, if I now downregulate microRNA, it should become a bimodal curve. That is what it is kind of saying right now. So the model kind of, I can make a sense out of it. Now, what happens for another cell line which is well known, if I upregulate, then cancer should not happen. Okay, that is opposite cell line. So in my MCF7, we know that if I downregulate, it will become much more cancerous. In MDMMB, it is well known that if you upregulate, then it will become cancerous. Okay. So what happens here is that this is MDMMB cell line, but this is we haven't been able to haven't done this. This is just like a D2E GFP reporter, not the 3' UTR. Even with that, we see is that it just shifts. It does not show a bimodality, even in absence of that reporter. Okay. So it seems, although we have a wild type kind of a thing, it seems that it is somewhere here. The curve is somewhere here, even in absence of microRNA. Okay. And the only way to get to a switching kind of a thing is to overexpress. So the idea is the following. If right now, it seems, although it's not this wild type thing, it is like something like that. Why I'm comparing this with MDMMB231? Because I know that overexpressing microRNA leads to cancerous state in MDMMB231. So idea here is the following. If I take, say, for example, a 5% peak, something like that, at any specific serum concentration, and I now start overexpressing microRNA, what I can see is the out of any one of these, like either it should become a bimodal or it should shift towards, like, if think about I'm doing it somewhere here. So then initially it is somewhere here, but now that I have overexpressed microRNA, either it will become a switch or it can go like this green line. If it is like green line, then the peak will just shift. If it is a red line, then this sort of peak should happen. So this is kind of a hypothesis right now in our mind. And the question is how I do this upregulation and downregulation of microRNA systematically. To do this, yes. No, we haven't come to up to that point yet. We haven't come to up to that point yet. Yes. That's the next step. That will be first. I at least see that whether my hypothesis holds or not. Okay, then we can see that. Network point of view, we have to do it experimentally. Network, though, we have already explained. That's it. With that model, you cannot explain how it is exactly. So you can actually make different microRNAs with a bulge and other things, and see that whether the extent of things are changing or not. So there are other ways of doing that. So a little bit more complicated. Right now, first prove the hypothesis that switch, I can move it. That is good enough for the right now. So what we are trying in case of MCF7, it will be something like this. MCF7, so already I have a kind of a, in presence of micron, I have a kind of a like this line, and then if I downregulate, I should go in a bimodal thing. So one case I have to downregulate, one case I have to upregulate. How I do that? To do that, we have made these two constructs. What are these two constructs? These are, if I give doxycycline, then I can induce or activate this reporter, and then I will be able to induce more microRNA in the system. That is the way to overexpress microRNA, if I want to overexpress microRNA. However, I have to quantify how much microRNA is getting produced. For that, our M cherry is also fused with that. So the level of M cherry will tell me how much microRNA has been produced, so that I can quantify that. Okay, so we have that already. We have produced this. We are producing this. These are anti-microRNA 20A. So these are kind of a microRNA sponge. So what it will happen is that if I produce this, it will act like a sponge and it will sequester the internal microRNA that is 20 is there in the system. So it will sequester that, and I will be able to downregulate microRNA systematically just by adding different levels of doxycycline. So this is what we are right now trying to achieve, and once we can achieve that, we should be able to do an experiment at a particular growth factor. So what we have showed earlier in that our theoretical model is that with respect to the expression level of microRNA, I can have a switch in E2F. That is what we predicted earlier. So right now what we want to prove is that by downregulating or upregulating to scan through this switching domain. But at a particular, I need to first fix at a domain where, at a domain of growth factor where I see a bimodality. If I see a bimodality at a domain of growth factor, then I will change at that growth factor, I will always keep that growth factor, I will change the level of microRNA, either downregulate or upregulate. Then I should be able to see that these two switches. That is my ultimate goal. So goal is little bit very difficult to achieve, but hopefully we will be able to, because all the clones we have prepared. Now what we are trying to figure out is that with respect to doxy, what is the level of, how I can systematically figure out the expression levels and put it in the model and figure out or fine tune our hypothesis. Then comes the questions of yours that how I say that these KDMP levels are different. So there we can make different microRNA probes, 20A, with bulges and this and that, and see that how they react in this. Like instead of making say this anti-microRNA, you can do different kinds of microRNA here, different kinds of sponges to change the probability or propensity, how they bind together. Mm-hmm.
In this figure or in the previous figure, you see that your bifurcation diagram becomes an irreversible bifurcation diagram.
Yes.
So in those cases,
if it is at all, but that was the we will make a model for this itself. We haven't manipulated that model itself. So you have already seen that with MCF7 we have made a different model already with the presence of RB. MDMMB also we have to play with little bit to see that how that model works, whether really I get a bimodality or not. That's the first thing by overexpressing. We hope. See, that feature will be if it is really irreversible, if I give a serum pulse, it will get stuck there. It will never come. That will be the experiment will tell you. that thing, if it is like that. Okay. So obviously exciting time is ahead of us. I don't know how much excited you people are, but I feel really excited because in a sense, at least one part we have already seen, that though I can down-regulate and I will get a switch. The other part is that whether I can overexpress in MDMA and get that thing done. That will be the coolest part. And then if we can somehow figure out whether that is the KDMP or something else, that is the thing. So this is kind of, we have already worked on, and we are working on this for other things also. So here, basically you, as I said, we have already seen that people have doing this work for a long time, that just the cells growing under the dish, and see them at two different ways, one at the population level and one at the lineage level. When I see them at the population level, I will see such kind of distribution of cell cycle time and G1 time and SG time, something like that. When I see them at the lineage level, I see these sort of correlations. However, nowadays, because of this Fouki probe, which is a fluorescence ubiquitin cell cycle indicator, this probes allows you to follow very meticulously that how much time cells are spending in the G1 phase, how much time cells are spending in the SG2M phase, and when exactly the cells are transiting from G to say S phase, because that will be given by a transient yellow color. And the way it works is that there is a protein called CDT1, which is G1 specific. There's a protein called geminin, which is SG2M specific, and they are actually controlled by two mutual inhibitors in the cell cycle. Like X and Y, they mutually are antagonistic to each other, and X and Y controls CDT1 and geminin via degradation. So what happens is that when X is high and X degrades CDT1, so Y will be, so geminin will be only present. But when Y is high, it will degrade geminin and only CDT1 will be present. So what they have engineered beautifully is that they have taken the degradation domain of the CDT1, the Japanese group, and fused it with M-cherry and given a CMB promoter, which is constitutively active promoter. The meaning is the following: if there is nothing to degrade the what is called CDT1, then the nucleus will always be red throughout the cell cycle, because it's under the control of a CMB promoter, which is the constitutively active promoter. However, there are people who will degrade CDT1, so you will see that color will go off. Similarly, they have put in an M venous with geminin, again the figuring out the degradation domain of the geminin, and they fused it again through retroviral transduction, and then you can get a cell line which will continuously give red, green, divide, and again become two red cells, and again continuously it will happen. So then you can precisely measure the G1 timing, SG2M timing, division timing, and so on and so forth, anything, and all the correlations and everything. Now people, we are not the first person. People have done this kind of work, and you can see that this was the paper I was talking about, that the kind of 2015 paper, which talks about that the circadian oscillation is the prime reason for doing this. There was another paper which talks about that because cells are spending a requisite amount of time in SG2M phase, that's why it is happening. Then there was a paper recently, 2020, from Thomas Hofer's group, my another postdoc supervisor. He said that nothing like that, this inheritance is completely deterministic factors dominated, and that is why it is happening. So point is, they are trying to figure out a very similar phenomena in different cell types, which everybody is seeing, but the observation is interpreted in a different ways, which normally happens in science. And we are no exception. We are also trying to do a very similar work, but from a different perspective. What is the different perspective? All these previous papers that I talked about, they only focused on the lineage data. They haven't focused on the cell cycle or the population level data. Like what happens simultaneously, these two together. What we tried is that, and not only that, they always did this experiment in an ambient condition, like 10% growth factor, where the cells grow very nicely. Okay, we created Fouki HeLa cells, means we take this Fouki, both these constructs, we put it inside retrovirally and make stable cell lines out of it. Once we make stable cell lines out of it, we can perform experiments at two different serum conditions. One is a completely healthy condition, one is a stressed condition, which is 2%. somehow kind of cells which experiences in a tumor environment. Maybe it's even lower than that, but a low environment where cells are stressed, but still growing. And we do a long time live cell imaging experiments for 72 hours. Now, I have to really gather a lot of statistics out of it. For that, what we have done, we have a rotating disc. What is that thing, Ganesh? Spinning disc microscope, and where the disc basically spins when you keep that your dish open, and then we can follow at 15-minute intervals in 15 different positions and take the acquisition very fast and then go to the next position, and that I can continue till 72 hours. Okay, and then I will get a lot of cell cycle related data, and we analyze that both at the lineage level and at the population level. And then obviously I have the expertise to make the mathematical model and make it a stochastic model, and we try to make a quite precise stochastic model so that we can really reproduce our experimental data and predict something out of it. That was the overall thing. Now, what we got out of it, that is the main thing. Okay. Now, when we do it for at 2% and 10%, we immediately see that at stressed condition, their cell cycle time becomes slower. On an average, you see that it is about 26.8 hours, but for the 10%, it is about 23.8 hours. So about 3 hours difference is there when they overall, on an average, they divide. Okay. And this difference is basically coming because the mean timing in the G1 phase has changed. In the 10%, the G1 phase is much faster, but in a 2%, their G1 phase is much slower. However, the SG2M time almost remain similar. When we looked into the coefficient of variation of the variances of this whole thing in a standardized variance, then we see that at 2%, the G1 variability is quite high compared to the G1 variability at 10%. Now, if you think of ideally, when I'm talking about something, if I want to use it in the say, for example, treatment purpose, in a tumor environment, I will have a very stressed condition. And under stressed condition, if my G1 variability is very high, that means not all the cells are committing for the cell cycle at the same time. And if my G1 variability is quite high, some of them will always escape my therapy. Because if the cells does not cycle, they will not pick up the medicine. So ideally what I want, a 2% like variability in a 10% like condition in G1. However, right now we have the opposite. The variability in 10% is lower, but the variability in 2% is higher. Okay, right now the data says. And not only that, initially we thought that maybe the G1 time and SG2M time is kind of correlated, but there is zero correlation. People have already seen that also. However, the interesting part is the following, and that we have seen in 4 different replicates when we did it both the times, all the times, is that when I switch from 2% to 10%, my cell cycle time correlates at 2% much strongly with the G1 timing. However, when we come to the 10% thing, the correlation pattern shifts, and the correlation becomes much more with respect to SG2M with the cell cycle. So these are my 4 critical results that I can say coming from just analyzing at the population level, not at the lineage level.
These are Pearson correlation?
Pearson's correlation, I think I squared.
Pearson correlation
Square.
So the Pearson correlation is actually like around 0.8 or
something like that. Now we move it ahead a little bit, and we try to make it a, like, if I look at the subpopulation level, say for example with respect to mean or median, if I try to segregate, what I see is that the slow cycling cells is kind of dominating the statistics, either of the cases. Not only that, as we observed in the experiment, the mother cells are much faster than the daughter cells normally, both at 2% and 10%. So these are something that we can immediately gauge out of it, and not only that, we can do a lineage level analysis, and what we found here does not matter is 2% or 10%, the daughters are always highly correlated, then comes the cousins, and then comes the mother-daughter, like what people have seen. So people have seen it at normal condition, we also see even at a stressed condition, that does not change. So we have all these data sets at the lineage level and at the population level. Point is, can any mathematical model make sense out of it? Okay. So we took that challenge. First, what we did, we make this like simple ODE based mathematical model. We try to get a saddle node invariant cycle bifurcation. Now you see the importance of the SNC bifurcation is that here now if I go from 2% to 10%, my period will only change 2 to 3 hours. If it would have been a normal Hopf bifurcation, in that 2% to 10% change, my period would have changed from 23 hours to 32 hours or something like that. So this allows you to constrain your periodicity and high amplitude oscillations, which is fine. However, nothing we'll be able to see because these are not stochastic model. So now I have to somehow make this a stochastic model, and I want to simulate this as we calculate data from a dish, like a single cell kind of. So I will assume in each single cell such networks exist. And what is a single cell to me? Say a random number generator, I pick up a random cell from a random number, and then from there I will try say that, oh, that was my first cell. Now for that first thing to first, I need all the set of parameters and the transcription rate. So all other parameters I kept fixed. However, the transcription rates, people have done beautiful experiments, have shown that the transcription rates for all the transcripts in mammalian cells are kind of log normally distributed. Like over the period and over the lineage, if you see, the transcription rates are always log normally distributed. So what we kept is that we took the mean transcription rate and created a log normal distribution out of those mean transcription rate, and have given them some variance of 5% or 10%, something like that, and then pick up one random transcription rate for each of the transcript and for our first picked up cell from a lineage. Make sense? So for that is my first cell with some random transcription rate from those log normal distribution for each transcript. And then we have already seen that in experiments also, they have seen that the transcription rates are kind of varying between mother and daughter in a very correlated manner, especially in the G1 phase. And the transcription rate variability can be from 10% to 80%. So we picked around 20% of the transcription rate for all the transcript for the log normal distributions. Okay. Then the other thing what people have seen is that, say in this Skinner paper and this paper, they have seen that the transcription rate gets modulated during the DNA synthesis and also during the M phase. So what we did, we started our simulation with Gillespie along with this transcription rate picked up from this log normal distribution. Then we reached at some point in the S phase, and then for each transcript we toss a coin because something is happening and some transcription rate either can increase or decrease. And depending on the toss of a coin, if it is less than 0.5 or greater than 0.5, we assume that the transcription rate either decrease or increase in a certain proportion. And the same thing happens at the M phase. Okay, again we talk, and in between we continue the Gillespie simulation, and at the end of the day, we either can divide the cells into half or into an unequal proportion. So we try to mimic as much as like for us possible to mimic the actual scenario, how cell divides and how cell go through during the cell cycle. So all these possibilities we incorporated in our model. Because what they are arguing, because of some epigenetic modifications, some of the nucleosomes gets crossed, so some rates can vary.
But the two daughters get the same transcription.
So that I'm coming. So the idea is the following. So when the mother cell divides, it faithfully gives all the transcription rates to its daughter. For the mother cell, when I'm picking it up randomly. But the daughters are getting from mother, inheriting from mother, and then they're going through the G1 phase, and around this point they get little bit different. Around this point, they might get little bit different, or they may again become similar. Because there is a random chance to become similar also. And then only this bit of time, the mother or that corresponding daughter, which is about to be dividing and becoming a mother, is spending with that transcription, those transcription rates, which it is giving to its next generation. So mother is spending a very little time with the transcription rates with which both the daughters are spending for the whole G1. That's one of the moral of the story we have to remember. Yes. Yes, yes. So for all of them, we have a different mean and the different variances.
So is this reported like?
Yes, yes. Yes, that is this paper. Nature Communication, Phillips. So you can I can share this. You can do it later. Okay, so I don't want to bother you with all the data, but the important part is that we played with this model for 1.5 years during the COVID period. Thankfully, COVID came. And we had only, my Vinodini can stop the experiments and only do the modeling. And because she can only do the modeling from somewhere from Coimbatore, and I was changing the, switching on and off the computers by going to the lab sometimes if we have chance. So she was able to perform these from Coimbatore with my advice from IIT Bombay room. Okay. And then we somehow managed to get all the features, not only the population level, but also the lineage level. How? That struggle I can tell you later. But we have done it. Now the question is that the things are coming pretty similar to what we observe in the experiment is not important. Why they are coming and what are the causal relations for the heterogeneity, that is the most important part. Okay? So now...
Sorry, so here I see that the cousin correlations are lower than the...
A little bit. Absolute things though is not possible. More or less. More or less cousins are a little bit less correlated than the mother. That is true, what you say. Other than that, data is right. That is true. That is true. I agreed. We admitted that in the paper also. Now the point is, my everything though is like what are then एक ही paper 50 साल चलेगा. So what we did is the following. Just to give you a nutshell. We started with a mother with all the deterministic network. We get the transcription rates for this mother from this log normal distribution. We simulated them with the GM, like till G1 phase. Until S phase comes in, we toss a coin, we change it again. Again we simulate it. Again M phase come, we toss a coin for each transcript, and then again simulate it, and we divide it. So this is my whole picture of the simulation. What we got this, right? Now I agree that we don't get cousins as like the experiments. I agree what you are saying. So this is all the results in nutshell. I'm going to now switch off one thing because it's a model. I cannot switch off. I cannot say that please don't transcribe this protein. If I say to one, the others will also not transcribe. But my model, I can do that. What we did is the following. First thing first. First, I say that, okay, I don't have any transcriptional fluctuation. All my mother cells in all the lineages are started with all the deterministic rates, deterministic transcription rates. Okay? So, what I switched off? I switched off the transcriptional variability in the lineage. I switched off the transcriptional modification during the cell cycle also. S phase, M phase, no transcriptional toss is happening, no toss. So I have intrinsic fluctuation, the molecular fluctuation. I have the unequal division, or the equal division does not matter. First thing first, you look at here. The lineage level heterogeneity that we used to see is completely abolished, more or less. So as soon as I switch off the transcriptional variability, I lose my lineage level transcriptional heterogeneity. Like all the mother-daughter. Basically, daughter-daughter correlation, little bit it is there at 2%, 10% is completely gone. All other correlations are completely gone. Not only that, the fluctuations levels have also gone down. The level of fluctuations that we used to see in the corresponding G1, SG2M, and overall cell cycle have also gone down. The correlation features have also have completely changed. Earlier, it was more G1 correlated and it was SG2M correlated. Now you can see in both of them it was G1 correlated. So all these features are completely gone when I switch off my transcriptional variability. So one thing I can definitely say, that transcriptional variability plays a very governing role to get the heterogeneity at the lineage level. Agreed?
So I'm not sure I completely agree.
Okay.
Because variability could be coming from many other sources.
No. Right now, from this model perspective, right now I can see that I switch off this, I don't see anything. That is excellent. That is exactly the point. Here, I don't know what is there any other English terminology? No, but this is no. But what he's saying is that, see, for example, if I introduce intrinsic fluctuation, I will lose my correlation.
No, but the correlations are built into the model via.
That is true. But see, for example, I have a deterministic model. Everything is 100% correlated. Deterministic model, everything is 100% correlated. Daughter, mother. Every cell cycle is 100% correlated. However, as soon as you impart an intrinsic fluctuation, you will see that the correlation will drop down.
So this is where I'm disagreeing a little bit. Everything is deterministic. There is no concept of correlation. Because
that is true. But if there exists, that is true. Everything is identical, right? But as soon as you impart the intrinsic fluctuation, you will see the molecular fluctuation in the model. You will see the correlations are very low. No doubt about it. You can try out 100 times. There is no doubt about it, you will see the correlation will drop down. Now the 1000 times question is this one, is that why on earth introducing another variability produces a correlation? Okay, we will come slowly, maybe. Now the point is the following. So here, this is, I switch it off, I see. Now the second switch off. What I want to switch off is that I will just take that each mother is picking up the transcriptional rate from a log normal distribution and nothing else. No in between cell cycle, it is not changing. Nothing is changing, and even I do not have any intrinsic fluctuation, like no molecular fluctuation. So what I am doing, I have a deterministic model. I am just simulating my deterministic model with some varying transcriptional rate for each cell. Then I see absolute correlation in sisters, cousins, and mother. I don't have molecular fluctuation, but I have fluctuation in the transcription rate from the starting mother, and that is given to the next daughter, to the next daughter, to the next daughter, and I see that everything is superlatively correlated, which is again not my experimental data. However, it is like that, and my coefficient of variations are also coming up. The fluctuations levels in the cell cycle at the population levels are also coming up, but not at the level we see in the experiment, but it is coming up. Okay. However, this is superlatively correlated. That also we don't want. Now, in cell cycle time, G1 time, SG2M time. So these are different colors. Everything is obviously cell cycle time is highly correlated, means G1 and SG2M has to be also highly correlated there. Otherwise, it's not going to happen. Now, what we did is that in the deterministic setup, no intrinsic fluctuation, I also incorporate these two fluctuations. So here I don't have my intrinsic fluctuation or the molecular fluctuation. I took my deterministic model. I first choose for different mother, different transcription rate. However, during the cell cycle also, the daughters are changing their transcription rate by tossing a coin. When I do that, I see I get back to the normalcy that the daughters are highly correlated and cousins or mothers are less correlated than the daughter pair. So what it tells you that internally when the cell cycle, during the cell cycle, when the transcription rates are varying, that allows you to kind of, at least in this model perspective, to locate that the cousins and sisters will be, cousins and mother-daughter pair will be less correlated than the sister pairs. Okay. Make sense? And obviously, when on top of that, you incorporated this intrinsic fluctuation, you get to this. So basically, intrinsic fluctuation is enhancing the fluctuations of what is available because of the transcriptional fluctuation and giving you the absolute variations that you are observing in the cell cycle dynamics. That is what we proposed here. But at the end of the day, what we want, what we get out of it? At the end of the day, what we want is that we have to somehow manipulate this. We can already see in that lineage level correlations are highly robust. At 2%, 10%, we are always getting correlation. Can I somehow manipulate my year level correlation, what is called the population level correlation? So here, what we have seen in the experiment in 2% and 10% is that what I was talking about, that the 2%, the 10%, the G1 variance was quite tight, but the 2% it was quite, wide. Okay, so and the point is, if therapy is working up to this level, I will have some cells escaping and they will relapse the cancer. Question is, can I make it even tighter so that everything falls into my cancer window, like therapy window? Okay, so for that, and obviously in 2% and 10%, this is the way my correlation varies at the population level. Can I switch it? Okay, to switch, what we did here, what happens is that comes as there's something protein called P38. So P38, what it does is a signaling molecule. Whenever there is a stress, at 2% there is stress, so there will be high level of P38. That we verified that P38 levels are high. What P38 is doing is making cell little bit nervous to go towards the cell cycle. So don't go for the cell cycle. So some cells are kind of reluctant, and because they are reluctant, you have little bit higher variance in the G1. So what we did, what was done to us, like COVID, people are dying, but don't let the other people know that people are dying. So you are in stress, P38 is high, and that's why cells are responding to the stress. So what we did, we give 2% serum condition, but with a P38 inhibitor. So there is stress, but we don't allow the cell to understand that there is stress. Okay. So what happens? First, we tried it in a mathematical setup. Obviously, what mathematical setup, modeling setup gives not always comes into the experimental thing. But what we can see that 2% with P38, I can see that my model is saying that not only the mean will shift a bit, and my variance will become tighter. What we found in the experiment, mean did not shift that much, variance becomes little bit tighter. Kind of, it is saying that hypothesis is not that bad. Maybe we can do something else also to make it happen. Moral of the story is that lineage level, and what we see that the lineage level variability is actually not changing even if you give inhibitors. It does not matter even if you do the experiment in the presence of inhibitors. So even if you change your network, some topology, something in the network, lineage level is becoming quite consistent, but I can change my population level heterogeneity. That is what we can actually achieve. So the moral of the story in our case, what we think is what is happening is that there is transcriptional variability at the lineage level, and if that is the only thing, then things will be absolutely correlated, almost highly correlated. However, because during the phases there is alteration in the transcription rate, that allows that sisters are a little bit more correlated, but mother-daughter correlations and cousin-cousin correlations becomes lower than the sister-sister correlation. And what happens here is that why sisters are more correlated than mother-daughter? Because mother is only spending 2 to 3 to 4 hours with the last set of transcription rate, with which sisters are spending about 10 hours in the G1 phase. And what we found, if you really look at the trajectory, so we simulated CDT1 and Geminin also. In our model, we found that only the Geminin, both the trajectories, in the SGTM, the trajectories have become different. The G1, they are kind of quite tight in the model. And if you look into the experiments also, see the G1 phase, cells are quite tight. So their dynamics is quite tight, with respect to which actually I'm measuring the G1 and SGTM. However, in the SGTM phase, they become different. So that PNS paper hypothesis that the SG2M time they spend equal, that's why they are highly correlated, is not that much correct. What we think is that here the G1, both the daughters are spending G1 with a very equal amount of like similar transcription rate. That plays a critical role. That is what we think is the right now things are, okay. This I will not bother you too much, just to tell you what we are doing in this third problem. This is an interesting, this suddenly came out of some work, is that what we were trying is that, again, we were looking at the dish with a Fuki MDM MB231 cells, this is not HeLa cells, and what we found that when we give them little bit of amount of TGF beta, we see that their cell cycle timings are kind of partly changing, but the dynamics, cells are actually, even without TGF beta, quite fast in the SG2M phase, which is kind of, again, counterintuitive. See, when we are young, we run very fast. I try to play cricket at this age. Right now, I can only walk this much only, because, older age, we become slower. Here, cells seems to be faster in SG2M phase. And when you give them TGF beta, they become even much faster than what you expect. The extent of increase is much faster, okay? Now here we don't go to the complete network model. What we try to do to understand these dynamics is to make an agent-based model. So we assume cells are agent, cells can be green agent or a red agent, and then there are 6 possibilities that can happen within that periphery, that cells can either go green, red can become green, they can move in either direction, they can die, they can divide, and they can, in the green phase also, they can move and they can die. So these are the full possibilities. And importantly, all these we can calculate in our experiments, every bit of thing. We put those data in those transitions and try to perform a simulation in terms of agent-based model. Believe me or not, we have not been able to produce the exact growth pattern. Then the question we ask that why it is happening. Then we see that the cells are actually kind of hand-holding and talking with each other. So they are mutually interacting. Now how I can impart the mutual interaction in my model? I can understand that cells can interact through integrins and other signaling. Okay, so here what we have done is that if this cell see more cells in this side, it will have a higher propensity to go towards that. Earlier, movement in all possible directions are equally probable in my model. Here now there is a probability that I will move towards the side where I have more number of cells, and that probability I can change mathematically time to time. And we see that around 0.5 we can actually capture the actual movement pattern that I observe in a particular growth condition. Now where I will apply that? So what we thought that we will do a numerical scratch assay. Have you heard of scratch assay where people have? So basically what they have done, they grow the cells to the confluence. Like the whole plate is full of cells. And then I make a uniform scratch in between of a certain length. Okay? And then I will allow the cells to grow and see that how the wound is getting healed. This kind of a wound healing kind of a model. So what we did, we got these parameters, we have these cells with these parameters, whatever exactly the agent-based thing, and then we started the simulation with this and tried to see that whether they will fill out the volume or not. What we found in the experiment is very interesting. Have a look at here. So we gave low amount of serum and then low amount of serum with a certain amount of TGF beta. You can easily see that here the wound is almost not getting healed, or cells are not coming. Here the cells are coming quite fast and completing the thing in presence of TGF beta. Okay. However, in 10% and 10% of TGF beta, there is a difference, but not that superlative difference. So what it means is that, and TGF beta has a very unique role. Low concentration of TGF beta allows cells to divide and allows cells to move. High concentration of TGF beta inhibits cell cycle and allows cells to move a lot. And that exactly we can kind of simulate also in our simulation profiles, where at the 10%, we assume that when I have 10% and TGF beta, I don't have any interaction kind of thing here. But here, if I add this much of interaction, I can actually, in presence of 10%, I can simulate the exact timing it takes to heal the cells. So here what happens is that in presence of TGF-beta, the cell-cell interactions become slower, and that's why the cells are populating that space of the wound much faster. So this is a kind of a work in progress. We have to do many other things and many other calculations. So I'm just trying to showcase that what different models can be done. So at the end of the day, I must thank all my team members. So this is Vinodini's work, mostly kind of what I showed you. She moved on to Cornell University right now. She's working with Tobias Mess in Lobis for the last 2-3 months. She's quite happy. And all the Kajal you can see. Ganesh is our active collaboration we have with Ganesh. And yes, this is my team. Thanks. Yes. Yes. Yes, this is exactly what we are trying to so we are going to do multiple replicates of this, and one of my so there's a MSc student, he's trying to see that if by analyzing these data, can I really segregate out what is the exact effect of only serum and the TGF-beta on movement and cell size. So I can actually track how many cells are dividing and moving, and how many cells are only moving and increasing their speed. So there is a way to track. So that confluent Fuki actually worked, Ganesh. That is doing. So there is a new paper came in from Israeli group, so which can actually track any kinds of this kind of a data beautifully, even without a H2B reporter, like even a nuclear reporter.
Just from bright field.
Yes. Bright field means with Fuki, but you don't need the H2B, like the blue, any nuclear reporter you don't need. And then you can do this scratch Asekar thing and everything. So by doing this, what we try to figure out is how many are dividing, how many are moving, how they are changing, and what is the exact role of growth factor and TGF-beta, such that at low concentration, why cells are doing EMT, can we get some hang out of it? So 2% and TGF-beta, we can see that 2% cells are actually not dividing. However, if you give them TGF-beta, they are dividing plus they are moving. That is exactly what we are trying to achieve. Let's see. There we have, yes, that we have again. So this student Pratik, Pratik is actually making a e-cadherin reporter, and with that we are also trying to tag cell cycle reporter. So that is a different work altogether. So people have seen that when cells are doing EMT, if you give them high dose, when cells are doing EMT, not only the G1S cells are getting G1 phase cells are getting stuck at G1S, even the G2M phase cells, which are at the mitotic phase, they are going back and getting stuck at G1S. So to figure out that, what Pratik is trying to make right now, a e-cadherin reporter, which will be a reporter for my EMT, kind of, and along with that a Geminin reporter, which will be a reporter for the cell cycle, and we have some plans for some experiments so that we can figure out whether really cells are getting stuck at G1S from either side or not, along with the models. So that is another thing that what we are trying to, what we can do here also. But MDMMB is not that kind, is already a kind of a mesenchymal cells. So we are right now trying this experiment with A549, which is more epithelial. Questions? Any other? Yes.
I'm still a little bit confused about the transcription rate.
Yes.
So what we have shown is that we had taken a much simpler model, right? Just a birth-death process for protein, RNA doesn't matter, right? Arbitrary. So as long as you have a birth-death process, so the protein or RNA is getting created with rate B and degraded with rate T, right? And this B can be exactly the same for all cells. Right? But just the fact that you have this stochastic process, and then you can give rise to daughters, you will get sisters that are strongly correlated, cousins that are less strongly correlated, mother daughters that are even less strongly correlated. So variation in the rates between different cells is not actually required. Same rates between different cells, but a stochastic process that will also actually give rise to the same phenomenon.
See, I have already shown you 5 other papers which have done give another. So exactly why it is happening? That is the $1,000,000 question, right? That is, see, we didn't figure out a way to vary the transcription rate as we want, because if I try to inhibit some transcription rate, it will affect many other transcripts. So right now I have no serious answer for this, to be honest.
There can be protein variability.
Right now you also cannot prove what you are saying. Yeah. So it's an open-ended thing right now. Let's see. Yes, that is already there in the form of Gillespie.
But that is not sufficient.
That is not sufficient. That is actually almost inconsequential for this lineage thing. That is only good for population level.
What is good for population? I missed the question.
Population level, I think the molecular fluctuation will actually destroy the correlation lineage level, if you only think of that.
He already has intrinsic fluctuation because of his birth in his model.
Yeah.
But that is not giving that
As much correlation that we want. That is what we have seen. But it will give at 2%, at a specific growth rate, it will give. At a different growth factor, it will not give. The thing is, when people did these experiments, they have only done in one particular factor, like particular serum concentration. There it will not reflect. As I showed you, if you do it in 2%, daughter sister seems to be correlated. 10% is not. So experiments are not done at different conditions. That was one of the problems. Yes.
we don't consider in a simple process. It's much complicated regulation.
And essentially, that will also vary from cell type to cell type. So right now, I don't know how to prove my hypothesis. I agree, because it was difficult experimentally. At least it's not my cup of tea.
Yeah, I think the only point I'm trying to make is that the simplest model where
And it works.
that itself will
Fine.
give, right?
Yes.
So when you make
See, if you look at the circadian thing, their model is like a cakewalk. But then the point is only that if you look into lineage, it will happen. Try to look the lineage data with the population level data and try to make a sense out of it. I can guarantee it will not happen. Because when you look into the particular kind of data, then you make a particular kind of model that works. Now you take 5 different kinds of experiments and try to make a sense out of it, which is happening at the same concentration that you are doing the experiment. It is going to fall apart. Anyhow, any model by virtue of its existence is wrong. Because if you change another thing, it will do another thing. So I am not defending my model. What I am saying is that it's a way of thinking. Yeah, I agree.
Any other questions? If not, let's thank Sandip once again.
Thanks.